Hello. All right. This should have subtitles coming on the bottom. There they are. All right. We can go now. So this is where we left glycosylation before. Pretty much everything we're going to be talking about, the antibodies and the spike protein, are glycosylated. The antibodies not so heavily glycosylated. The spike protein's very heavily glycosylated. So, quick little review. Remember we got our two types of glycosylation. Yeah, I bet you've got that. We've got our N-linked, which is linked by asparagine, which happens to have the code N, and it's also linked by a nitrogen, which also is an N. So, yeah, either way you slice it, it's definitely N-linked. That's the amine that's up here. And the other one is our O-linked, where we're linked by an oxygen, and both serine and threonine have a hydroxyl, which is an OH group hanging off the end. And so that oxygen is going to become the linker. It's going to shed its hydrogen, and it's going to bind onto the end of the sugar chain, which also has an OH on it. And so OH and H are going to leave, and they're going to become water, and you're going to be left with a sugar attached to the protein. Neat. Okay, so how does this happen? This is a good question. So we're talking about this little tiny thing up at the top. It looks like the Holy Grail from the Indiana Jones movies. This is called Sec61. Sec61 is this thing. This is the oligosaccharyl transferase. So oligo means a few. Saccharyl is sugars. So these are chains that are a few sugars long. And anything that ends in ASE means it's an enzyme, and then whatever comes before that tells you what it does. So this is a transferase, so it transfers oligosaccharides, little tiny things, onto something. And the something turns out to be all kinds of different proteins. So that's not too bad. This is going to happen at the ER membrane, the endoplasmic reticulum. And all the sugar chain addition is going to happen actually inside the ER. So this colored rounded rectangle over here, is the inside of the endoplasmic reticulum. And so first of all, we're going to have this oligosaccharyl transferase, this OST, stick the oligosaccharide onto the protein that we're making right there. Fantastic. The protein's coming through Sec61. That's right. It's the thing that pulls the protein through the membrane. And so, then there's going to be various steps down here. Now, the interesting thing is that there is going to be a folding checker. And one of these is going to be this BIP. And essentially, if one of these folding checker proteins is able to bind to the protein that's just been made, then that means it's not very well folded, and it needs to either learn how to fold itself better, or it needs to go and get recycled. And so you see the BIP over here, it's recognizing it and saying, oh, that's not great. And so if that helps us fold up, then fine. Or if not, we're going to send our protein down here. And calreticulin and calnexin are also going to try to help fold this protein up. They're going to help sort of crunch it down so it folds up properly. And if it comes out folded, great. It'll then get exported. But if it is not folded properly, then it goes up into here. And it's going to go toward the disposal pathway. But you've got a bunch of different proteins here, EDEM proteins, and they are going to bind to it. And there are a couple salvage pathways along here. We can try to trim the glycan, make it a little shorter, see if that works. Try it again, see if it'll fold up. Or we can try to extend the glycan a little bit. And yeah, if those don't work, then fine. We're just going to bind it. This BIP is going to check it again, make sure that it's terrible. If it is, then fine. We're going to drop it off, and we're going to take it out and shred it. So it's not going to go out the normal way. So good proteins go out toward the Golgi inside of a little vesicle. So they're on the inside of this little membrane structure. And these are going to all blob together, and they're actually going to form the Golgi complex. Because the Golgi is not so much a thing, it's a place where membranes are constantly coming in and merging together to make these little flat pancake-like stacks. And then membranes are constantly going out the top side. And so these are going to come into the bottom of the Golgi and form part of the Golgi, and then eventually move through it. And I'll show you what they do when they move through the Golgi. So here is the Golgi apparatus. Marvelous. Don't you agree? And in the Golgi, what we've got are a lot of ways to both trim down the glycan and add on to the glycan. So you've got a bunch of these things called glycosyl transferases. And so instead of calling them oligosaccharide transferases, we're calling them glycosyl, but oligosaccharide and glycosyl mean about the same thing. So they got different names that kind of mean the same thing. They're little proteins that are buried in some part of the membrane, and some of these things tend to float in the early Golgi. Some of these tend to float and hang out in the late Golgi, and actually have other proteins that will retain them there. So the other proteins that come into the Golgi will move through the Golgi. These guys are kind of stuck there, and so they'll get left behind. And each of these is going to add on a different little group to the end of the glycan. And so you've got extensive trimming of the glycan. That's essentially what the Golgi is for. We're going to trim that glycan down, and then we're going to cap off each of the ends in a slightly different way. And there's a lot of complexity. There are a lot of different things that could theoretically happen, but only a few that actually do happen. And so you end up with pretty consistently glycosylated proteins. They'll be all pretty close to one particular glycan structure on one particular part, which is very interesting, the way they're able to end up like that. And so this will be characteristic for the protein. That is the idea. And so this is where that diagram comes from, which is actually a very nice review of how the Golgi works in glycosylation. So since we're dealing with glycoproteins, I thought this is useful. I hope it was. Thank you very much.